welcome to the stage ISCB President Thomas Lingauer and ISCB Fellows Chair Bonnie Berger.
Good morning. The first off is we'd like to recognize those members of our society, the ISCB Fellows for this year, who have achieved the most and should serve as role models for all of us. The first fellow up is Vineet Bafna. I don't know, Vineet? Okay, that's okay. But there's Vineet, and he's from UC San Diego, and he works more on the algorithmic end of computational biology. Elazar Eskin has let me know he's running his summer course right now because it was scheduled before he found out, but he will be here next year, and we can hopefully let him pick up, or at least stand with the other fellows. But who is here is Shirley Liu, who I'd like to come to the stage. And
congratulations. And there's a pin for you. I wanted to fool myself trying to pin that on you.
I have mine on, by the way. Every fellow gets a fellow's pin, which I have on my lapel, and Shirley just got hers. And she's at Harvard School of at the Dana-Farber, Harvard School of Public Health, exactly. And the final one is Marie-France Sagot, who has given so much to our field on the theoretical end. So let's give a hearty round of congratulations to the new ISCB Fellows.
Now welcome to the stage ISMB ECCB Conference Co-Chair Torsten Schweid.
Hello, good morning everyone. As the local conference chair, it's my great pleasure to welcome you on behalf of the Canton of Basel, the University of Basel, and the Swiss Institute of Bioinformatics to ISMB ECCB 2019. We're very excited of hosting you here in Basel, and I hope you will enjoy the scientific content of the conference, but also a little bit of the city, despite of it's going to be rather hot in the coming days. But we have a river which is nice in the evenings. Already now, ISMB ECCB 2019 is one of the most successful editions of this conference series. We have more than 2,000 registered delegates from more than 60 countries. We have an outstanding scientific program with 6 keynotes and 625 oral presentations. Now, if this large concentration of science makes you a little bit confused, there is a conference app which you can download for your mobile phones, either Android or Apple, and that helps you to navigate the program so you don't miss out on anything important. Now, just in case you still miss out on an important talk, what's different at this conference is all the keynotes are streamed online, so you can watch them later on the ISCB YouTube channel or ISCB TV channel, and also all the presentations will be recorded. So if you want to go back and revisit a certain topic because you missed it here on the conference, no worries, you can do this later. One important part of the conference are always poster sessions, especially for younger scientists to showcase their latest research and to discuss their latest work. Please, I would like to encourage you, use the opportunity, talk to the poster presenters, discuss their work, but also to all the poster presenters. There's a new feature in ISMB this year, which is an unconference-like feature. So if you would like to give a flash talk about your poster, please register. There are 60 slots available. for our CompBio Ignite theaters to give a quick presentation, 5 minutes, either on Tuesday or Wednesday. Now, of course, what makes a scientific conference successful is the quality of the science. And this all depends on all the colleagues who have spent a lot of time over the last months in making sure that the scientific program is what you're seeing here today. Many people have worked very hard as part of program committees, as reviewers, cozy leaders, peer reviewers, organizers of tutorials and workshops. Can I please ask everyone who was involved in one way in organizing the meeting, as a reviewer, as a chair, as an organizer, to quickly stand up? Thomas, you did something. Thank you. Because these are the people that are serving our community, so we can all enjoy an exciting and enlightening conference. I wish you all an enlightening ISMB ECCB 2019 and like to hand over to Ron for the introduction of the morning keynote. Thank you.
Please welcome ISCB Awards Chairman Ron Shamir.
Good morning. I chair the awards committee, which makes the recommendation on the 4 highest awards given by the society, and the awards are finally approved by the ISCB steering committee. And this is an opportunity to urge you all to submit your nominations, because the starting point of all the process is nominations from ISCB members. And today I'm happy to introduce our winner of the Innovator Award, Bill Nobel. The Innovator Award is a mid-career award for outstanding contribution for scientists who are up to 20 years from their PhD. So Bill grew up in Illinois. He had parents who were college professors, but he says he did not have a really scientific inclination at the time. Nevertheless, he went on to Stanford for an undergrad that combined symbolic systems and philosophy, graduating in '91. An unusual piece of personal history is that he then went on to Lesotho and did 2 years in the Peace Corps, teaching science and math to secondary school students. And in the evenings, he started getting interested in programming, so as long as electricity was around, he would do some coding. In '94, he went to UCSD for a graduate school, and together with his mentor, Charles Elkan, he developed HMMs, hidden macro models for protein and DNA sequences. His first bioinformatics article was on the web server for motif finding, MIME, which is still in use today, quite remarkably. After graduating in '98, he went for a postdoc with David Hausler at UC Santa Cruz. And there he also did the first. He co-authored the first application of SVM to gene expression. And the same period, he also developed new kernels for joint analysis of heterogeneous, what we now call omic data. I don't think the term existed. In '99, he joined Columbia University, and in 2002, he moved to his current position at the University of Washington, where his main position is in the Department of Genome Science. Since then, he also expanded his interest to unsupervised machine learning methods for gene annotation, machine learning methods for proteomic data, and also analysis of high-C data for understanding chromosomal architecture. Bill has published a remarkable number of more than 230 papers to date, and even more importantly, supervised nearly 40 graduate and postgraduate students, many of whom are now independent researchers. He won the Postdoc Mentor of the Year award of the University of Washington. This is an award given by the Postdoc Association, so this is dedication to education is quite remarkable. Bill is an active member of the ComBio community. He serves on several editorial boards of journals. He's on the ISCB boards and on various committees. Please welcome Professor Bill Noble.
All right, thank you very much. This was quite an honor and a surprise when I initially got the email from Ron informing me of this award. And I hope that I can give you a talk that gives you some idea of the kind of stuff we're up to. And it's a little daunting to be dubbed an innovator, because now I have to keep on innovating in the future, but I'll try. As I thought about what to talk about today, or do I need to do something here? Somehow I have to get to my slides, is that right? There they are. Coming. As I thought about what to talk about today, I would like to say that a few years back I had this grand vision that I was going to work on moving data between different mathematical spaces, but it's really more of an after story. I came up with some stories I wanted to tell you, and then noticed this common theme. The background for my title slide sort of implies Chewbacca hitting the hyperdrive to go into hyperspace, but really it's about not, if you're in hyperspace, you go from one place to another in 3D. And I'm going to talk today about different ways to move from one dimensionality to another, and hope to convince you that this is a useful operation when you're moving either to higher dimensions or to lower dimensions, if you do it in a way that's informed by machine learning. So it's three different stories that I'll tell. Each one has a name that I'll explain as I go along. The first one is going to be mass spectrometry, which I know most of you probably are not so familiar with, so I'll try to give you a little background there. And then the other two are genomics, which I think is a more commonly studied theme here, both a single cell and a bulk genomics project. So I'm going to start off with the first one. Gleams is a recursive acronym. Gleams is a learned embedding for annotating mass spectra. And this is work that was done by a very talented graduate student in my lab, Damon May, along with sort of our partner in crime, Jeff Bilms. He collaborates on, he's a machine learning expert in electrical engineering who collaborates on a lot of our projects. So as I said, I'll give you a little background first on mass spectrometry. For those who aren't familiar with it, tandem mass spectrometry in particular, as applied to proteins, is sort of the most high throughput way to identify and quantify proteins in a complex biological mixture. The main problem that people doing mass spectrometry face is that proteins, that's very dim. Proteins like those guys in the upper left corner are very complex 3D molecules, and they don't behave very well biochemically. And so when you do mass spectrometry on proteins, you actually don't analyze intact proteins. You first digest them into peptides using something like trypsin, and then you run them through a mass spec device. So here's a picture of an older mass spec device in my collaborator's lab, Mike McCoss. It's actually a tandem mass spectrometer, so it does actually 2 rounds of mass spectrometry. But for the purposes of this talk, the details of that are not so important. The main point is that it produces a lot of data. You run one of these for half an hour or an hour, and you'll get tens of thousands of observations. Each observation is a spectrum where the horizontal axis is the mass-to-charge ratio and the peaks correspond to intensities or abundances of different fragments when you've taken a particular peptide and broken it into pieces. And what we've spent a lot of time over the past decade doing is applying a computer to these data and trying to figure out what is the peptide species that's responsible for generating each observed spectrum. So Damon had also spent a long time thinking about this problem, both in graduate school and before when he was working with Marty McIntosh at the Hutch. And he was interested in addressing this more general question, which is, can we try to stop thinking of every mass spec experiment as if it's the first one that's ever been done, and instead try to analyze mass spectrometry data more jointly? So in particular, mass spec has been moving forward over the past 10 years or so to sort of catch up with genomics in terms of making data publicly available, so that now it's very common for most mass spec journals require that if you submit a paper to the journal, you also have to submit your data to a public archive, much the same way that goes on with the short read archive or GEO in genomics. And so there's these different public repositories that are available for that. And the unfortunate aspect of some of this is that often what happens is that you do your experiment, you generate your data, you analyze the data, you write your paper, and then, there's another one. Perfect. Thank you. You write your paper, and then just before you click submit, you deposit your data into a repository so that you can, follow the rules and get it all publicly available. And what we like to encourage instead is that people deposit their data earlier on, because they hope that by depositing their data into one of these archives that they can then get back information about the experiment that they performed. So this is not, of course, a new idea. Many people have thought of this kind of thing before. There are existing repositories that make sort of variants of the repository publicly available for use in analyzing your own data. You can download, for instance, something called a spectral library from various repositories. And some of these are automatically generated by clustering either previously identified spectra or clustering all of the spectra. The question that we wanted to address was, could we exploit peptide labels during the training of one of these large-scale models of tandem mass spectrometry to hopefully achieve a more robust, efficient, and interpretable model? And so the idea that Damon set out to test was, rather than doing a fully unsupervised clustering, we would try to train an embedding into a latent space in which all of these spectra reside. The idea is that some of your spectra will have labels associated with them because they've been identified previously, and some don't have peptide labels associated with them. And if you then see a new spectrum, you can embed it into this space and then very efficiently look for your neighbors in that space and try to figure out something about maybe what was the peptide species responsible for generating that spectrum. So as with a lot of machine learning projects, one of the headaches of this was just trying to figure out the best way to actually get the data into the machine learning system. Mass spectra are a little bit finicky in the sense that they're really kind of bags of peaks where each peak is an intensity and a mass-to-charge ratio, and the actual precise value of the mass-to-charge ratio is really important. And so Damon spent quite a bit of time experimenting with different kinds of representations, and we ended up with kind of a mixture of different ways to represent the spectrum. We have some parameters that represent the experiment, properties of the experiment itself, some that represent observed properties of the intact peptide, so its total charge and its total mass, and then multiple redundant ways to represent all the fragment information, either by discretizing the mass-to-charge axis and then counting peaks that way, or also computing a similarity, a high-resolution similarity to a set of reference spectra. So with this representation, we then trained a machine learning system to learn an embedding that would go from a high-dimensional representation of a spectrum to a lower dimensional space in which all the spectra would reside. So here's just a sort of an overview of the kind of network that we're talking about. On the right here is a picture of the network. It's a somewhat deep neural network in the sense that it has several layers. It's not like 50 or 60 layers like some of the really deep networks. For those who aren't familiar with deep learning, I've got some terminology defined over here. There are some components of the network, like convolutional layers that are designed to identify particular features within parts of the input, and then fully connected layers that simply give a complete connection from one layer to the next. And the main thing is that you have the different sets of input features segregated and being processed separately, and then concatenated together and put through a fully connected layer with an output dimensionality of 32. The dimensionality of the space in which the data resides is itself a hyperparameter of the method that Damon experimented with quite a bit. Now, I told you the idea here was to try to exploit peptide labels during training, and it's not obvious how you would do that in this kind of network. What we actually do is embed the network I showed you on the previous slide twice into this kind of architecture. This is a commonly used architecture in things like natural language processing called a Siamese network, where you have two copies of the network. They have tied weights, so any update on the left also affects the network on the right. You can put in pairs of spectra, and those spectra then get pushed through the two networks, and their locations in the embedded space give you an update according to this loss function. So you basically have a label associated with your peptide that indicates whether, or it's associated with a spectrum, indicating whether the two peptides are the same or different. And then you're using the Euclidean distance in the embedded space. And there's two components here. The first component basically tries to say if the two peptides have the same label, I want them to be as close together as possible. And the second component says if they have different labels, I want them to be far apart. But if they're farther than a particular pre-specified margin, then that's far enough, and I don't have to push them any farther than that. So Damon assembled a collection of about 5.6 million spectra from on the order of 20 different experiments spanning a broad range of different machine technologies, as well as different kinds of samples, different species, even including meta-proteomic data, and then trained a network. So here's just a visualization. This is using TSNE to visualize the 32-dimensional space in 2 dimensions. And you can see there's some structure here. Just to give you an indication of what's going on, Damon randomly chose some charge states and canonical masses, and then colored all of the dots that had that particular charge and mass combination. And what you can see is that all the dots of a particular color tend to co-localize in this 2-dimensional space, implying that sort of the large-scale structure in the embedded space reflects what you'd expect, the charge and the mass properties of the intact peptide. If you then were to zoom in on, say, for example, this region here, you actually see when you zoom in closer that there is more structure there. So these are just different peptides within, or different spectra within that circle, and they segregate by their precursor mass. But of course, the question is, what is this bias? What do we gain by doing this embedding? And so one of the things that we looked into was approaching that idea that I outlined on a previous slide, which is, if we see 2 spectra that are close together in the space, maybe we can use that information to help propagate labels in the space. So this turns out to be, it can be quite computationally expensive to do a clustering algorithm. And also, we're calling this community detection rather than clustering, only in the sense that the clusters that we aim to find are quite small. So we're kind of under clustering the space. There are plenty of spectra that will be in their own cluster by themselves. But if we see lots of spectra very close together, then we want to be able to say, okay, these are probably all generated by the same species. So we came up with a straightforward, what we call a hub and spoke method. We use, the first step is finding the k nearest neighbors of each spectrum. We use a library that was developed by Facebook called FAISS, F-A-I-S-S, to do this step because it can be done quite efficiently. We found 1,000 nearest neighbors, mostly because you want as many as you can get, but 1,000 got to be a little bit expensive, so that was as many as we wanted to find. And then we do a simple iterative procedure where you essentially sort all the spectra by how many neighbors they have within a particular distance, and then sort within those by the mean distance to their neighbors, and then go down the list and sort of greedily assign each spectrum to its own cluster and grab its neighbors. So you say, essentially, okay, this is my first guy. He becomes the hub, and all of its neighbors within tau become spokes. And you go to the next one, and so on, until the very end when, because of this 1,000 nearest neighbors step at the beginning, occasionally you get two clusters that are close together, and those get merged in a post-processing step. So we wanted to evaluate how well this would work. We looked at essentially a space of how many communities do we have that have just a single peptide label in them, and how many communities have multiple peptide labels, and what you'd like is to be sort of in the upper left corner here. We did a comparison to several different standard clustering algorithms. One of them, for example, is k-means clustering, which turned out to be quite expensive for this kind of application, and also tended to, regardless of the value of k, end up very far away from the ideal point in the upper left corner there. The hub and spoke method, by contrast, put us over in this range. We ended up choosing a tau value of 0.095 because it gave us about a 1% false discovery proportion among the detected communities. This was a reasonably fast thing to do. As I mentioned, the k-nearest neighbor part with the face library was the most expensive step. So embedding actually only took about 20 minutes, 19 minutes for these 6 million spectra. Then the k-nearest neighbor, and then the hub and spoke method just takes a few minutes. At this stage, we can look at how well we do in terms of increasing the number of detections made by the algorithm. And so if you look at the 1.6 million identifications that were available just from the original database search, we can boost that by about 8%. Which in this area is a fairly significant increase in the detections. And I should mention that boost comes from two different ideas. One is the kind of label propagation that I mentioned. If you're near to a peptide with a label and you don't have a label, then you can propagate that. But it also comes from the idea that if you see sort of dark matter, that is a big cluster of spectra, none of which is identified, you're pretty sure they're all detections of the same species, but you don't know what, then you can afford to spend additional time analyzing that with more intensive strategies that would be too expensive to do on the entire compendium in the database. And so we did that, and those two strategies together gave us the additional identifications. This would correspond, if we had done this, for example, to all of the data in the massive repository, getting about 15.3 million additional spectra. And one thing that was interesting is that if we looked at the remaining dark matter, those things that we spent extra compute on and still couldn't identify, and used some fairly standard quality measures, the ones that were unidentified seemed to be lower quality, indicating that they might be contaminants. So this was sort of a proof of principle. What this plot shows is just what proportion of your unlabeled spectra have a labeled neighbor as a function of the size of the set of spectra that you embed. 5.6 million is actually quite small compared to the full compendium. And so the goal as we move forward is to scale this up and put it into production mode so that we can learn one of these embeddings from a large collection of spectra and then automatically put new spectra in as we go. Another direction that we're investigating is whether or not, once we've learned this embedding, there may be important semantic relationships. Those of you who are familiar with natural language processing applications of these kinds of ideas have seen this concept. Word2Vec and other kinds of learned embeddings have this property that has been noticed where you can do vector operations in the embedded space that capture semantic relationships among the embedded words. The most common examples is this idea that you can take the vector representation of king, subtract man, add woman, and end up close to the vector representation of queen. And so the question is, could we also find similar kinds of semantic relationships between, for example, different charge states of the same spectrum, or different post-translational modified forms of the same peptide? So that's story number one. That's the mass spec stuff. Now I'm going to switch to a different story quite different in the sense that we're now doing single-cell genomics. And here, most of this work was done originally by Jia Liu, who was a postdoc in my lab at the time. He then left for a faculty position at University of Michigan, and so the work was completed by a graduate student in his lab and another postdoc, Ritambra Singh, in my lab. And it's a collaboration with Jean-Philippe Vert, who is a longtime collaborator of ours who's currently at Google Brain. I probably don't need to convince people of this, but it does seem increasingly clear how important it is to integrate different kinds of genomic data, and in particular in the single-cell genomics setting. So if you imagine that you have a population of cells, you can split it into aliquots and subject each of those different aliquots of cells to a different single-cell genomics assay. So you might measure gene expression with RNA-seq, or open chromatin regions with ATAC-seq, 3D structure with Hi-C, or even just do microscopy, make pictures of the cells. Most of these, with some exceptions, it's not possible to do co-assays to measure the exact same feature in the same cell. So instead, you get different measurements in different cells. And so that's one of the things that makes this whole problem challenging. So I'm going to start off with kind of an example to give you an idea of the kind of problem we're interested in. Imagine that you've done single-cell RNA-seq, for example, on a differentiation process. So you have maybe multiple time points, or actually cells at different points in a differentiation process. You run some trajectory reconstruction. This one happens to be monocle. And you get some kind of branching structure, which maybe even agrees with your prior intuition about different fates for this particular population. You then do single-cell ATAC-seq on the same cells, but not exactly the same cells, different aliquots of cells from the same populations, and you see a similar bifurcating pattern. The question is, are these cells down here, are those the same cells? Or maybe it should be that the correlation goes this way or this other way, or who knows? So the thing that makes this difficult, well, one of the things that makes this difficult is that we don't necessarily have a one-to-one correspondence between cells, and we certainly don't have any information about which cell is which in the two different experiments. But also, there's no real one-to-one correspondence between features. You could imagine in this setting with ATAC-seq and RNA-seq that maybe you could come up with some correspondences based on if a gene is active, then it might have open chromatin regions near it. But we want this to work even for things like single-cell Hi-C or even single-cell images where there wouldn't be any correspondence between features. So the kind of methods that we're interested in are methods that integrate single-cell data by projecting to a shared manifold. And so the idea would be you take your two sets of cells with N1 and N2 numbers of cells in each one. You run them through some manifold alignment algorithm. And then embed them into a d-dimensional space. And hopefully, if you do this well, then the two structures that you've started with get superimposed correctly in the common space in which all of the cells reside. In order for this to work, it has to be the case that there is some common manifold structure that the learning algorithm is able to pick up on in the data. This, again, is not an entirely new idea. Other people have applied manifold alignment algorithms to this kind of data. So, for example, there's this paper from 2011 from Wang et al., which basically involves constructing a joint Laplacian across the multiple domains and then doing an eigenvalue decomposition, followed by a sort of k-nearest neighbor graph to characterize the local geometry in the embedded space. There's also the Guma algorithm, which is a generalized unsupervised manifold alignment algorithm. It's kind of similar to ours in the sense that there's an explicit objective function that's being minimized and with different components of the objective function. One of the challenges for Guma compared to what we're interested in is that Guma makes the assumption that there is a one-to-one correspondence of cells between the different domains being aligned, which clearly won't always be the case if you have different kinds of single-cell data. And then there's also the Megan algorithm, which is based on actually a pair of generative adversarial networks. And And this is basically learning an embedding, or actually two different embeddings in a sort of competitive way. The Megan algorithm empirically seems to work best if you have some, actually works best if you have some correspondence information about the features. So when we approached this problem, it was actually Jean-Philippe who pointed us to this MMD measure, which stands for maximum mean discrepancy. This is a general measure that was introduced in the machine learning literature about 7 years ago now by Andrew Gratton and some collaborators for characterizing how similar two distributions are in a given space. It's characterized in terms of kernels, where a kernel is a generalized notion of similarity in a given space. And so you can think of the MMD as characterizing, if you think of the similarities between cells within, say, an RNA-seq sample would be in this yellow, all the similarities between cells in an attack-seq would be in this area here, and then what we're hoping to learn is a single matrix so that we can also compute similarities between RNA-seq cells and attack-seq cells in our example. And all the MMD does is basically have different components corresponding to different parts of this similarity matrix so that you get these two, the blue parts, to be large and the other parts to be small. So you, on the face of it, it seems like all you need to do is basically say, we're going to take our two, or actually it could be even more than two, generalized to any number of data sets, project them all, and then just minimize this MMD so that we're showing that they have the same overlapping distributions in our embedded space. And so this is just visually what we're hoping to see, and you were doing a minimization over the parameters of our projection so that we minimize the MMD. Turns out that's not sufficient by itself for a couple sort of obvious reasons. One of them is that it's really easy to project things so that they fall on top of each other if you say project all the data to the origin, right? It's not an interesting projection. So you have to have something in there that sort of penalizes some trivial solutions. And so that's what this second term does. And then the third term, actually, we also think it's really important, of course, that when you do the projection, the data in the embedded space resembles the data in the original space, right? You don't want to just completely throw out the structure of the original data. And so this third term here is essentially akin to what you would do in, for example, multidimensional scaling, right? That you say, I've got a similarity matrix in my original single-cell data. I embed it over here, and I'd like the similarities in the embedded and the original space to be as close together as possible. And then there's some hyperparameters that trade off the different strengths of these different terms. So we started off by simulating some data to demonstrate that it was possible to do this in principle. So we take an original manifold, we sample some points, we do a random projection plus some Gaussian noise. So here's the sampling in 2 dimensions, and then we do a random projection to 1,000 or 2,000 dimensions. I can't really draw those, so that you just have to imagine that those boxes are very high dimensional spaces. And then we do the MMD-MA algorithm, this manifold alignment algorithm, to try to recover the original structure. We intentionally misspecified the dimensionality of the embedded space because, of course, you wouldn't know ahead of time what dimension the manifold has. But even when you have a 5-dimensional manifold and a 2-dimensional original space, you can show that the structures get recovered. We did a couple other simulations. Here's one called a circular frustum in 3D. The term frustum was new to me, but apparently it essentially just means this kind of a helical structure. We used this in part because we wanted to try to think about situations where you have a cell cycle component to your data as well as a differentiation process. And so you can see the 2 different samples from a frustum getting overlaid here. We actually used the simulations to quantify how well the algorithm was working. We could compare it to the JLMA algorithm that I mentioned, using either 5 or 6 nearest neighbors. This is just the fraction of samples that are closer than the true match for each given cell in the embedded space. So ideally, everybody's closest neighbor is the correct neighbor, and you'd be at 0. And you can see that the MMD-MA is doing quite well in all 3 of these simulations. We then went on to look at some co-assay data, because it allowed us to validate that the method was working well. So this is data from this Nature Methods paper in 2016, where they're doing a co-assay between RNA-seq and DNA methylation. So this is data from the same single cell, where you have the gene expression and the DNA methylation. And the idea is we're not going to tell the algorithm which cell is which. We're going to allow it to do the alignment, and then afterwards check whether or not it successfully aligned the DNA, the expression data, and the methylation data from the same cell. So here's what happens if you take the data. This data set had about 62 cells in it. If you take that data and just project it using PCA to a two-dimensional space and overlay them on each other, and as you can see, each cell is pretty far from its corresponding neighbor. After you run the MMD-MA algorithm, the lengths of these connecting edges get shorter, so every red dot here is a gene expression measurement, and the blue dot that it's connected to is the methylation measurement from that same cell. And you can quantify how frequently the guy that you're connected to is your nearest neighbor is the correct nearest neighbor by looking at this distribution. This is just showing among all the cells what proportion of the samples are closer than the true match. So about 50% of the cells have as their nearest neighbor the correct nearest neighbor, and on average, it's only 2.4% of the cells are closer than the true match. So currently what we're doing is moving on to looking at things like aligning single-cell Hi-C and imaging data. This is work that was done by a rotation student in the lab, CXQ, where we were taking data generated by the 40-nucleome consortium. This is single-cell Hi-C data, and imaging data from the Allen Institute for Cell Science in Seattle. And the idea is that we'd be able to superimpose the two sets of measurements. We're particularly interested in pulling out components of the data that correspond to the cell cycle. And so that's what CX did, was try to find ways to extract features relevant to the cell cycle to allow these to be superimposed on each other. Those of you who aren't familiar with single-cell Hi-C, the concept is that you have a matrix representation of the genome, where each entry in the matrix corresponds to the two different loci in the genome, and it's a count of how close together those two loci are in a population of cells. In single-cell Hi-C, you have many very sparse matrices, and so we have this very sparse representation, which we wanted to try to use to capture cell cycle information. This is fairly well established in the literature by now, is that you can see cell cycle information in this kind of data by looking at the distribution of counts as a function of distance between the loci that are in contact. So this is a figure from the first single-cell Hi-C paper from Nagano et al. And what you can see is that there's a strong diagonal, and then there's an increase in long-range contacts at different stages of the cell cycle. And so what CX did was basically take marginals along the diagonal here and use that, this is called the contact density profile, and use that vector as a representation of the single cells. This is a standard method that's been used before, and you can show that this very quickly recaptures, so this is a UMAP embedding of those vectors, and that recaptures the cell cycle information. On the imaging side, we were able to extract, again, from the images provided by the Allen Institute, 4 different features that they told us are highly correlated with the cell cycle, corresponding to surface area and volume of the cell and of the nucleus. Again, you get a UMAP embedding here, which is suggestive of the cell cycle. And so we took these 2 data sets, projected them onto each other with MMD-MA into 10 dimensions. And this is just a visualization of what it looks like in the shared space according to UMAP and PCA. And it does look like they have been successfully superimposed. On the other hand, the cyclical structure is not visually evident. It wasn't clear to us whether this was a problem. Of course, we're projecting to 10 dimensions, and this is just a 2D representation. So to quantify what was going on in the embedded space, we basically did a nearest neighbor analysis, where we find the nearest neighbor cell image for each single cell Hi-C data cell in the data, and then use the labels from the single cell Hi-C to look at the measurements from the corresponding neighboring images. And that's what you see in this slide here. What you can see is that the surface area and volume features are following what you'd expect in terms of trends for the different stages of the cell cycle in the images that have been aligned to the high C. So that's where we're at that stage, and now I've got a little bit of time left to tell you about the third project. Now I'm going to switch to bulk genomics in the context of the Encode Consortium. So probably many of you are familiar with Encode. Encode stands for the Encyclopedia of DNA Elements. It's a consortium that's been active since 2003, and my lab's been involved since the beginning of Encode. Encode is essentially a data generation consortium. Initially, it was a technology development, but these days it's largely devoted to generating data about the human genome, the human epigenome, the human transcriptome. So there are various kinds of measurements that capture things like gene transcription with RNA-seq, or protein binding with histone modification ChIP-seq or transcription factor ChIP-seq, and then DNA local packing density with things like ATAC-seq and RNA-seq. Encode, like the roadmap consortium from which this figure was taken, carries out these kind of analyses in many different cell types and tissue types and so on. In Encode terminology, those are all called biosamples. And so what you end up with is a measurement along the whole genome for every combination of biosamples and assay types. So here, for example, is a short region of one particular part of the genome where you have 3 different biosamples, HSMM, MSC adipocyte, and so on, and then 3 different histone modification measurements. You can think of these as vectors, in principle vectors of length 3 billion for each of your high-throughput genomics experiments. One of the challenges that we face is that doing all of these experiments is just simply not feasible in terms of the compute time, or sorry, not the compute time, the expense in terms of sequencing, and also just the human effort involved. So here is just a snapshot of part of the matrix. This is just for histone modifications and DNA successibility, and a set of biosamples. And you can see that there are some that are quite well characterized, but there are many missing data points. And so one of the challenges that we've been interested in is, can we predict what would happen if you were to do a particular genomics experiment before it actually was done? So the work I'm going to tell you about was done by Jacob Schreiber, who's a grad student in my lab. He's also here somewhere, although I haven't seen him yet. Also precursor work done by Tim Durham, another graduate student in my lab, and again in collaboration with Jeff Bilms. And I have to mention the software that implements this is called Avocado, and everyone wants to know why it's called that. It's just because Jacob likes to name his software after fruit. As far as I know, there's no other reason. And this image is supposed to be an avocado with some missing data. We started working on this problem quite a while ago, and it was motivated in part by the Netflix challenge, which many of you will be familiar with. The Netflix challenge was essentially a matrix imputation problem where Netflix made available a bunch of data about viewers and movies or TV shows, and they gave you preferences. You know, these individuals rated this particular movie between 1 and 5, for example. And the idea was, could we predict how well this particular viewer would like this particular show? So methods that worked really well in the Netflix challenge were based on matrix decomposition, where you decompose each show or movie into a latent representation, and each user also has their own latent representation. And then, if you say, you know, for example, this component in the latent space might correspond to whether or not this show is thought-provoking. And then you ask, okay, does this particular viewer like to think? Well, if they do, then maybe they'll like that particular show. Conversely, the next one might be, does this show have Adam Sandler? Does this particular person like Adam Sandler? And so on. So here's an example that Jacob came up with showing that he really likes the show Gossip Girl. I'm not going to comment on that, just he does. And the show factors are shown here in this rainbow pattern here. And the main idea is that you have some kind of function that says, oh, look, it's the same rainbow here and here. And so therefore, probably they're going to get a high prediction for this particular show. The original work we did was by, as I mentioned, Tim Durham on this model called Predicted, which was published in Nature Communications in 2018. And Predicted essentially generalizes from that 2D matrix decomposition to a 3D tensor decomposition So now you have components in the latent space corresponding to genomic positions, a separate latent space for the assay factors, and then a latent space for cell type factors. And then you can do a kind of generalized scalar product between these three different latent representations to get a prediction for the particular experimental measurement that you think will happen if you do this assay in this biosample at this particular genomic location. What Jacob did was generalize this a bit further to replace that function f from being a linear generalized scalar product to being itself a deep neural network. So this has the nice property that it can capture nonlinear relationships among the different factors, and it also allows us to have different latent dimensionalities of the different spaces that we look at. So there's, the assay, genomic position, and cell type factors can have different dimensionalities. We spent some time in the paper describing this, validating that the avocado predictions made by this deep tensor factorization model work well. There's just visually to give you an idea, here's some data from chromosome 1 for H3K4ME3 in a particular biosample. And Chrome Impute was the very first method to address this problem. This was from Jason Ernst and Manolis Kellis. Chrome Impute used a collection of random forests to make these predictions, and it gets a mean squared error at this particular locus of 0.2867. If you look at predicted at this locus, you get a drop in mean squared error down to 0.09823, and then we get an additional improvement at this locus down to 0.0933 in the avocado predictions. We were particularly interested in this paper in understanding which of these predictions are easy and which ones are hard to make. Intuitively, if you think, well, let's imagine that your training set has, say, 14 different biosamples for a particular assay, and at every one of those biosamples at this particular genomic location, we see a peak. Well, it doesn't take a rocket scientist to think, well, maybe in the 15th one, you're probably also going to see a peak there. And the same is true vice versa. If you sort of group regions of the genome based on how many times they have peaks, the things that never showed a peak in the training set are probably not going to have peaks in the test set. And so what we did was we grouped the genome according to how many times you saw peaks and then evaluated the mean squared error separately in each of those. So we compute the mean squared error within each group. And that's what's shown in this plot. You're seeing the mean squared error on the vertical axis here for a particular mark. And this is the number of cell types in which those regions are a peak. And you can see that you see the expected trend. The easiest ones are things that are never a peak or always a peak. And in the middle is where it gets the hardest, but consistently across all of these, the avocado model is giving an improvement. Recently, Jacob has expanded this to move to the full Encode data set. In the original paper, we used the same exact data set that was used in the Chrome Impute and predicted papers so that we could do a fair comparison. But now we've expanded, so going from 127 to 400 different cell types and from 24 to 84 assays. And in particular, we've expanded to include transcription factor binding, which the other models weren't attempting to do. This is a much more challenging task in general. As one comparison, here's a comparison to the recent Encode Dream challenge. So these are all the competitors here, and what you're seeing is a precision recall curve, and the line here is the line of equal precision and recall, and the performance measures are the average precision and the equal precision recall point. The top line here is sort of the best you could possibly do. This is the true signal for this particular experiment. And the second ranked one here, at least at the EPR point, is the avocado model. I should point out this isn't a totally fair comparison. On the one hand, against avocado, all the other models actually have nucleotide sequence as part of their input, whereas we don't. They're working in a different genome build, so we had to do a liftover step, and they're also working at a broader resolution, 250 base pairs, whereas we're doing it 25 base pairs. And then on the other hand, avocado has many more input data types available to it. So it's, again, it's not a totally fair comparison, but the point is that in terms of the state of the art of making actually accurate predictions of TF binding, if you want to use these predictions in your research, we believe that avocado is the state of the art. Here's just another example, a very different kind of factor, rest in liver, and again, similar kinds of trends. But one of the things that I wanted to emphasize here is that although this model was initially designed as a way to impute data, actually in the back of our minds what we were thinking was what we really care about, or at least another thing that we care about a lot, is that maybe the latent representations themselves are useful. So we thought if you could take, for example, in the avocado ENCODE model, there's on the order of 10,000 different measurements that go into this training of this model. And maybe the latent space that's been learned there can be used for other tasks. So to give a qualitative sense of whether this hypothesis might be true, here's just a visualization showing the different kinds of latent embeddings. Here's genomic embeddings. We've just embedded some promoters and some enhancers, and you can see that there's a separate promoter cluster, a separate enhancer cluster, and then this mixed cluster of sort of potentially bifunctional locations. The assay embeddings show similar trends in the sense that if you look at all the, say, histone modifications that are transcription associated, they reside close together in this embedded space. The activating ones also show a clumping here, and the repressive ones are together as well. And on the cell type embeddings, again, you see things like embryonic stem cells and induced pluripotent stem cells close together, various blood cell types, and so on. So it looks like, qualitatively, maybe there's something interesting going on in all three of these embedded spaces. So what we thought was maybe we will focus in particular on the genomic embedding and ask whether this representation of the genome could be used in other downstream tasks. So imagine you're going to try to make some predictions along the genome. We wanted to compare different kinds of inputs that you could give to a machine learning model. So you could imagine taking just the data from ENCODE for a particular cell type or biosample. You could imagine taking the full set of imputed data for that same one, either from Chrome Impute, Predicted, or Avocado. You could take the latent factors for that particular region from either Predicted or Avocado, or sort of as a baseline, which I don't think anyone ever does this, but you could try to just take all the data that's available for that particular position and use all of it as a predictor. We took all of these different possible inputs and just put them through a standard machine learning method, in this case a gradient boosting classifier, and then tried it on a variety of downstream prediction tasks. In each case, we did a sort of leave one out chromosome by chromosome prediction and just measured how well we could do in terms of average precision. So the first task we looked at was predicting gene expression. What you see here is just cell types sorted by the baseline accuracy. The baseline is just predicting the average expression of that particular across all the training set cells. And so it's very hard to see all the dots here. So what I've done is given you some summary statistics here. In every case, in all the cell types, the avocado does better than if you just used the data directly from ENCODE. Across all 47 cell types, and you get an increase of 0.144 average precision. And even in the more difficult ones over here, you get an even bigger increase in average precision. If you compare to using the full roadmap compendium, which is the second best method, it's still working better to use the avocado latent factors in 36 out of 47 cell types. And the other methods fall sort of in between in terms of how well they work. We applied this as well to predicting physical promoter enhancer interactions. We used a data set from the Whalen et al. Paper called Target Finder paper. There's some details there that involve fixing up that benchmark because there's been a published critique that shows there were some challenges associated with putting that benchmark together. So I'll reassure you we've tried to fix those problems. And what you're seeing here is just the average precision for predicting physical promoter enhancer interactions, again, for all these different measures in 4 different cell types. And the tallest bar here in at least 3 out of the 5 cases is the avocado latent factors. In this case, you have the predicted latent factors doing a little bit better. Actually, it's 4 out of 5 that or no, 3 out of 4. And you can also see that avocado does reasonably well as well in many of these cases. There are several other places several other tasks that we've looked at. These are described in more detail in both the original paper about avocado as well as the follow-up that generalizes to the full ENCODE compendium. You can get the model itself and all the source code from GitHub. And what we're doing currently is putting all of this on the encodeproject.org webpage. So currently, if you go to encodeproject.org, there are 3,048 imputed experiments. These are from the predicted paper, so you can use those directly in your research. We're in the process of uploading the more than 30,000 experiments from the avocado predictions, and those will also be available on the portal soon. So those are the 3 projects, and I thank you very much for your attention. I'd be happy to take questions. Do you want me to call?
Thank you for a wonderful talk. I wanted to ask how feasible you think it is to use the avocado models to predict the effects of perturbations or treatments or disease.
So are you thinking of like single nucleotide perturbations?
No,
Or
the
what
effect
kind
of a
perturbation?
treatment on...
Oh, I see. So the challenge with avocado currently, because it's set up as this tensor factorization, is that you need to have some measurements in both states, right? In other words, it's filling in a tensor. So you can't really use avocado to predict in an entirely new row or layer of the tensor. But if you had some measurements in the perturbed state, then you could predict the other measurements. In my talk at the MLCSB on either Wednesday or Thursday, I'll talk about how to choose which of those measurements to do in that
Forward
perturbed
to
state.
that talk then. Thank you.
Bonnie,
Excellent talk.
thank you.
Really interesting and broad. I was wondering what the runtimes of some of these algorithms are, because in my understanding, nearest neighbor, is close to quadratic runtime.
I missed one word.
Nearest neighbor is close to
Oh.
quadratic runtime, even with some optimizations.
So are you talking about in particular the
Even the
first?
single cell, like just doing nearest neighbor, or even the first application.
So the first application, that was definitely the nearest neighbor was the most expensive bit, right? It was like 20 minutes to embed everything and 15 hours to do it. And that was for a relatively small repository. In that case, it was partly driven by the fact that we were trying to get our approximation as good as possible by looking for 1,000 nearest neighbors. Clearly, if we scale up to a much larger compendium, we're not going to do 1,000. We might have to do 10 or something like that. But I agree with you that the scaling in the embedded space, that's an ongoing, it's challenging for almost any of these kind of embedded applications, the k-nearest neighbor stuff.
Thank you.
Any other questions? So one question about the MMDMA. You mentioned in brief that you, in principle, could apply it for more than 2 types. Have you tried this? Does it scale up?
So we haven't, I don't think we've done it in simulation, which is what we should have done first. We kind of jumped to doing it. We actually happen to have a data set where we have single cell RNA-seq, Hi-C, and ATAC-seq. And so because we had collaborators who wanted to see that, we put them all in and ran it, and it didn't seem like it made sense. But then we backed off and just did 2, and even there it was challenging for that particular data set. So it's not clear yet whether that was because of the generalization to 3 data sets or because the data was challenging. I think what we should do, and what I'll try to ask Ratambra to do, is simulate in 3 data sets. We haven't done that yet.
Hi, Bill. Really nice talk. So I was wondering, when you were trying to look for your 1,000 nearest neighbors, have you tried RPCL, where you can define, say, rival penalized competitive learning instead of I think you tried k-means, is that correct?
So all we've done so far was use that library from Facebook, this FAISS library, to find the k-nearest neighbors, because that seemed like probably a state-of-the-art method, but it wasn't something that we didn't evaluate any other approaches for that, so I'd be interested to hear about alternatives.
All right, thanks.
Okay, one more question? Yes, please.
Oh, there's one there. Oh, I don't know who's good.
Thank you very much for the great talk. I'm wondering whether the fact that avocado is not using the sequence information, is that by choice? Because the sequence information might give you overfitting, and you're in principally using the behavior similarity across the genome coordinates?
Yeah, so it is by choice. It was mostly that it was the simplest thing to do was simply look at the data that was in the ENCODE project. But conceptually, it's not challenging, really, to generalize to including DNA sequence features. You would have to use some kind of maybe a one-hot encoding and then cope with the fact that the DNA sequence is invariant across all the different biosamples. So there's some tweaking of the training of the method, but it is something that's on Jacob's to-do list. I'd be very interested to see what happens, especially in the transcription factor binding setting.
Hi. Thank you for the wonderful talk. I just thought I'll ask a question regarding Gleams. So we mentioned that quite a bit of the spectra are not identified. I know it's a common thing. I was wondering if you looked into the ones that had decent quality, but they were clusters that were annotated to see if they have post-translational modifications on it.
Yeah, so I mean, that was essentially our strategy, was to take all of the large clusters with no identifications and then do more intensive search strategies. So that involved exactly that kind of thing. One is relaxing the trypsin or the digestion conditions, so you allow more missed cleavages and so on, but also searching for common modifications. And so that most of those things went into that 8%. There are still many large clusters of unidentified things that don't obviously show a post-translational modification and can't be identified. And this is an ongoing issue in the field as a whole, is do these correspond to other kinds of unexpected contaminants, for example, or different kinds of fragmentation patterns. And that's an open question for the field.
Thank you.
Okay, let's thank Bill again.